All right, welcome everybody to our Cold Climate Fruit Production webinar series. My name is Amaya Tucha and I will be today's webinar moderator. Today we will be discussing understanding cold hardiness and bud break in grapevines, insights from temperature variation and modeling with two great speakers, Dr. Jason Londo and Dr. Al Kowaleski. I'm going to just read a little brief bio from our speakers. Dr. Kowaleski is an assistant professor in the Department of Plant and Agroecosystem Sciences at UW-Madison. His research focuses on how woody perennial plants survive extreme conditions, particularly winter bud survival, using approaches that range from gene expression analysis to phenological modeling to uncover plant resilience mechanisms. And Dr. Londo is an associate professor at Cornell University and specialized in fruit crop physiology and climate adaptation. His work explores how apples and grapes and other fruit crops respond to climate stress, with an emphasis on winter hardiness, root-stock-scion interactions, and sustainable solutions for fruit production. So together, Dr. Kowaleski and Dr. London bring a complementary expertise on plant resilience and climate adaptation, offering us insights into how perennial fruit crops can thrive within environmental challenges. So we're very excited and thankful for both speakers to be here today. And just some housekeeping issues, please. If you can stay muted and turn off your video, that would be great. If you have questions, feel free to add them into the Q&A or the chat box. We will be answering and reading through the questions at the end of the webinar. And so with that, I would let Dr. Jason London start with his part of the webinar.
Okay, I'm going to grab the screen. And just to check, can you see it in full view or is it stuck in presenter view?
Full view. Looks
okay,
perfect.
And you can see my cursor as well?
Yes, we can.
Okay. All right. Well, thanks, everybody. Thanks for the introduction, Amaya. My name's Jason. I'm working here at Cornell looking at grapevine cold hardiness, and I get to introduce some more of the context and background information for this overall webinar. So I'm going to be focusing mostly on the processes of acclimation and deacclimation and some of what we've learned studying this in grapevine. And so to begin, we're just going to start, of course, with our main character, Vitis vinifera, the European grapevine. It's the dominant species used for producing grape products worldwide. Something like 95 to 97% of production comes from this one species. It's responsible for all of our really nice end products, wine, juice, table grapes, and raisins. It was domesticated from its wild ancestor, Vitis vinifera subspecies sylvestris, from the Mediterranean basin. And in the context of winter temperature, the Mediterranean region, pictured here in this little sort of mock-up of minimum winter temperatures, you can see that that region is quite warm. So the species is adapted to a Mediterranean climate, which is typified by cool, mild winters and warm, dry summers. And even though much of its range doesn't have a lot of strong freezing conditions, it's able to survive some pretty impressive winter temperatures. It can survive down to around -18 to -25 Celsius or -10 to -13 Fahrenheit, depending on the cultivar. And we contrast vinifera with wild North American grape species, or the diversity of North American wild grapevines. And so wild Vitis can be found throughout the United States. Pictured here on the bottom right are 4 example species. The lime green is the county level distribution of those species. And you can see that there's overlap between these 4 particular species. And they range all the way from the Gulf Coast up into Canada. When we look at the cold hardiness that wild Vitis can manifest, they can survive winters down to -32 to -40 degrees Fahrenheit. So some really, really deep cold temperatures. These species don't produce very nice fruit, but they can be used for cultivar improvement, and they're key to breeding new varieties to handle diverse climates. All right, I'm sorry. I'm not sure if you guys are seeing all this other stuff, but it's super distracting. So in the context of the US, where we try to grow grapes in this country, of course, our climate is much more challenging than a Mediterranean climate. So pictured here on the left is the north central US in 2012 and 2013 for the hardiness maps, and the northeastern US on the right-hand side. And what I want you to take from this set of figures is two things. Vitis vinifera is limited to growing in basically the 6A to 6B region, which is the Kelly green region, and hybrids can be grown down to around 4A, which is the magenta region. What you can also see is as we've moved in the last decade from 2012 into 2023, is that our hardiness zones are shifting. And that's primarily being driven by changes in our winter temperatures. And so regions that couldn't grow vinifera before can grow vinifera now, sometimes. And regions where it was just too cold even for hybrids, those regions are also being opened up. There we go. And so, as I just hinted at, winter is warming. And in North America, winter is actually warming faster than any other season. And so in the picture in the top left corner of the screen, you can see the, I don't know what the geographic distribution is of these little squiggly lines in each state, but you can see that in the north central and northeast region of the United States, that winter's warming the fastest. Since 1970, 5 to 6 degrees Fahrenheit. And pictured below is just a minimum temperature graph showing you how the minimum temperatures of winter are decreasing overall from 1970 to 2020. Like I said, almost 6 degrees of Fahrenheit. What this means for our overall climate is a little bit messy. We know that winters are warming, and we know that as the polar regions of our planet warm up faster than the equator, that we have changes to the jet stream. And the jet stream is really our best friend as it comes to cold temperatures during the winter, in that a strong jet stream helps contain polar air up at the poles. A weaker jet stream tends to allow for a lot more wobbles. And this manifests in the now frequently stated polar vortex, and which you can see pictured on the bottom right of the screen is an example of the 2014 polar vortex. We just had one of these a few weeks ago. I think for those of you who are listening in the central United States, you may be experiencing some of this right now. Okay, so that's the framework that we're working from as far as what our climate is doing. So now let's go into a little bit about how this all manifests with cold hardiness with grape. So when we talk about cold hardiness, what we're talking about is the ability to survive freezing throughout the entire winter without suffering any damage. So pictured on the right is in red and blue highs and lows of a given year, November through April on the x-axis. And if we say something that is cold hardy, we have this cartoon of this U shape. And basically, if the minimum temperatures of winter don't intersect with that dashed line, we would consider that variety to be sufficiently cold hardy and escape freeze damage throughout winter. As I mentioned, the shape of this response is a U. The beginning of this response is acclimation, where the tissue is gaining cold hardiness in response to decreasing temperatures. In the middle of winter, they maintain that hardiness. And then as we enter into late winter, early spring, and temperatures begin to rise, the tissues that we're talking about begin losing their cold hardiness in a process called deacclimation. Key to this whole process is that the plant is dormant. And so if the tissue that we're talking about, and in the case of grapes, we're talking about dormant buds, if the bud hasn't gone into dormancy in the fall, it cannot acquire cold hardiness. At the beginning of winter, the buds enter a type of dormancy that we call endodormancy, the beginning part of dormancy, where growth is being suppressed by some sort of internal physiology. And what that means is they're very resistant to heat. So if you go out to the field and you collect a bunch of buds during the endodormant period and you try to get them to grow in a greenhouse, the bud stays sealed up, you don't get any growth. Now, at some point during winter, this type of dormancy transitions to another type of dormancy called ecodormancy. And in this stage, instead of physiological suppression of growth, we have temperature suppression of growth. What that means is those buds are very responsive to heat. And if you go out and collect them from the field and put them in a greenhouse, you'll get them to break quite rapidly. We don't understand entirely how the buds transition from endo to ecodormancy, but we know that they're paying attention to something called chilling hours. And this is simply the amount of time that the tissue spends within a certain temperature bracket. In this case, in this example, between 32 Fahrenheit and 50 Fahrenheit. Every hour in that range, they sort of count and add up. And when they reach enough chilling, then they transition between these dormancy states. The important part to this is that you can transition between endodormancy and ecodormancy because it's based on temperature exposure during winter at these low but non-freezing temperatures. You can transition at different points between different years. So that brown bar may transition to the green bar earlier in some winters or in some regions. It can also transition later. All right, so these are the interacting factors that are overlaid on how our cold hardiness changes through winter. We can track where dormant buds are using a technology called differential thermal analysis. And we'll just call it abbreviation going forward as DTA. Grapevines use this very particular mechanism called supercooling to suppress freezing to very, very deep temperatures. As I mentioned in the beginning, vinifera can go down to negative 13 Fahrenheit. Hybrids can go down to negative 40 Fahrenheit. We're not going to get into what supercooling actually is, just that we can exploit this mechanism to measure the point at which buds freeze. And pictured here at the top is a top-down view of a compound grape bud. The primary bud is at the center. This is where their floral tissue is. Secondary and tertiary buds are backup buds that are typically less fruitful. The primary bud tends to be the most sensitive. We can assess cold hardiness through DTA. What we do is we remove these buds intact from the cane. We place them in these thermoelectric plates. We create this freezer sandwich where we put a bunch of samples into the freezer and we slowly freeze them overnight, measuring the voltage within these thermoelectric plates. And the idea here is that when the dormant bud freezes, it emits a puff of heat, and we can detect that puff of heat in these thermoelectric plates. The data, the rough data, just in case you're curious, looks something like this. This is just voltage as temperature goes from the freezing point to negative 40 Fahrenheit. We get this large spike in voltage. This is the freezing of high temperature exotherm, or this is the water that is between the bud scales. This is harmless freezing. But then as temperatures decrease, you get these 5 little spikes, and these are 5 independent primary buds freezing. We measure these independent spikes and take an average to determine the temperature that kills 50% of our buds. And then we use that metric, the LT50, as a way of comparing between different cultivars and different years. That data looks like this. Here's 2 years, 2018 and 2019. The black lines are the weather conditions. In blue is the hybrid variety La Crescent. In pink is the vinifera variety Cabernet Sauvignon. And you can see from between these two different curves, you got that U shape. The shape differs between years, and that the hybrid is more cold hardy, particularly at midwinter, than the vinifera variety. We know that maximum hardiness is driven by the pattern of winter cold events. If you don't have a very cold winter, you get a shallower curve. So if you have a nice cold winter, you get a nice deep curve and a lot of separation between vinifera and hybrid varieties. If you have a mild winter, there's less separation because these buds on these grapes are sort of lazy. They don't achieve a maximum hardiness unless they are pushed by the climate that they are in. And that's important as we think about how cold hardiness changes as our winters become more mild. When we look across different varieties, here I'm ranking vinifera and hybrid varieties from weakest at the top in red to strongest in the bottom in blue, freeze resistance. And so here broadly, you can see that viniferas are the least cold hardy, and I've placed some temperature ranges on the right in case you're interested. Hybrids tend to be most cold hardy, and then you've got this slushy middle where you've got overlap between the two gene pools. What we see here is that while each year has its own personality, the rank order of these different varieties is roughly maintained. Viniferas are typically always more cold hardy than hybrid varieties if you look at the average year. But also that cultivar response is dynamic across the season. And you can see that with some of these varieties where you have red and blue switching positions, meaning they are more sensitive or more hardy than the mean. So what that goes to tell you is that these different hybrid varieties and vinifera varieties are using the different patterns of winter to exhibit different levels of cold hardiness. So there's a strong genotype by environment interaction. All right, so that's cold hardiness in general. Now I want to zoom in a little bit on acclimation. And so what we've learned about acclimation is not a whole lot. It's a very tricky thing to study. When we look at acclimation curves or the beginning of the cold hardiness curve from field data, we see that there's some things that pop out. Early in winter, there's a general conserved acclimation between vinifera and hybrid varieties in that they're responding to decreasing temperatures in a similar way. The slope of these two lines is roughly similar. But once temperatures get close to freezing, we see this divergence where both vinifera and hybrid varieties begin gaining cold hardiness, but hybrid varieties use every temperature stimulation as a little bit more efficiency. It gains cold hardy faster for each little trigger. The other thing we see is that once they reach maximum cold hardiness, the same type of temperature exposure, these low temperatures here, don't seem to trigger additional cold hardiness. So what we think this means is that for each sort of temperature dose that the grapes get, they have a response that they can muster, but they require increasing doses to continue gaining cold hardiness. We also have learned that it's not just the minimum temperature that is important. So in this experiment, what we did is we looked at Cabernet Sauvignon buds, we collected them from the field, and we placed them in different growth chambers. And I've pictured the colors up here at the top, 36, 45, and 52 degrees Fahrenheit. If we look at cold hardiness in the field in the black line, you can see it decreasing here. Our experiment, we don't see any decrease in temperature, even when you take buds that are warm or cold, no difference in response. But if we cycle the temperature between this minimum and the maximum, we're able to trigger cold hardiness across, regardless of whatever temperature they were experiencing previous to this. And in fact, what we see is that there's a 16-degree oscillation in this experiment, and that causes a 9-degree acclimation response, at least of Cabernet Sauvignon. What I'm not showing you here is we've done some other preliminary work, and larger oscillations and colder oscillations tend to drive the cold hardiness deeper, but we don't have it really dialed in beyond what I'm showing on this slide. And the take-home point from this is that the pattern and cycle of fall temperatures seems to drive maximum hardiness potential. It remains unknown to us what's going to happen if falls continue to get warmer before the entrance into winter. So that's what we know about acclimation. I want to quickly mention that we are also trying to understand how dormancy is changing throughout the winter because we want to understand the relationship between cold hardiness and dormancy. And we typically do this through forcing assays. These are very simple. It's like the example slide I started with in the beginning. You grab a bunch of buds from the field, you put them at room temperature, and you measure how long until they break bud. So here on the y-axis, days to bud break, time of winter along the bottom. And what we're really looking for is a transition from insufficient chilling, where bud break is very sporadic or non-existent, to full chilling, where they are all synchronized. And this is a measure of that transition from endodormancy to ecodormancy. The way that we use forcing assays is a little wonky, in my opinion, because we just arbitrarily pick a date where we have enough synchronicity and an early enough bud break to say that's enough chilling. In this case, we're choosing the cutoff of 28 days or 4 weeks. So if you put the buds in warm temperatures for 28 days and they reach bud break, we say that's enough chilling. If you draw that line here with this data, you can see that La Crescent requires less chilling than Cabernet Sauvignon. What we've done in our work, and this is actually Al's work, and so I'm stealing the thunder from him, is looking at how deacclimation can be used to measure this transition from dormancy in a more accurate way than just the forcing assays. And so the concept here is to take cold hardiness of field material and see how fast they lose their cold hardiness when exposed to warmth. So here's some temperature data. Chilling is accumulating from left to right. We go out, we collect field data, and we see what the LT50 is. And then we collect extra buds and put them in a growth chamber and see how resistant they are to losing their cold hardiness. And here in this example, when we look at the slope of that line, here is time in days in a growth chamber. How much cold hardiness do they lose over time? And what we see is they lose about 0.1 degree Fahrenheit per day. Now, as chilling accumulates and you go through winter, you gain cold hardiness. You do this experiment again, and now when you deacclimate them, you see that that rate has gone up sevenfold, 0.7 degrees Fahrenheit per day. You finish that out at the end of the winter with maximum chilling, and we see that we almost double that again, so now 14 times, 1.3 degrees Fahrenheit per day. The cool thing here is if you map this deacclimation rate data with bud break data, like I showed you in the previous slide, you see this nice little correlation that early in the winter with low chilling, this would be endodormant, that buds take a long time to break and they have a very low deacclimation rate. But as they gain chilling, they get faster at both. So if we look at this throughout the whole winter, what we see here is deacclimation rates for Cabernet Sauvignon for every week of the winter. You can see the size of the line is the speed. And so what I want you to notice here is that the line gets steeper and steeper as we go through the winter, and even more so in Le Crescent. And if you remove the differences in cold hardiness and just look at the rates, you can see that both varieties are getting steeper and steeper throughout winter. If we look across all of our varieties, we see they all have the same response. So this response is very conserved. And to make this easier on the eyes, what we do is we just convert these rates into this curve. So here, deacclimation rate per day on the y-axis and chilling hours on the y. So time of winter on the y and how fast they lose their cold hardiness. I'm sorry, I just muddled that. Time of winter along the x and how fast they are losing their cold hardiness when exposed to heat on the y. And you get this nice S-shaped curve. I've shown you where Le Crescent and Cabernet Sauvignon are. Again, what we're tracking here is the change of endodormancy into ecodormancy as manifested by changing rates. If we look at the range of variation across those varieties, what we see is here, most hybrids, in particular, hybrids that come from cold hardy hybrid breeding programs that use Vitis riparian and Vitis labrusca, they tend to deacclimate very fast. That's the top of this range, up into the 4 and 5 degrees per day. Viniferas are in the middle, and then hybrids that come from more southern species like Cinnaria, rupestris, and estivalis, they tend to be slower. However, if we want to really understand how dormancy is changing through winter, we don't want to look at these maximum rates alone. We want to normalize each cultivar to itself. So if you take this 100% point for each cultivar and normalize the data, what you get is this much flatter S-shaped curve of normalized deacclimation potential. All that you really need to know about this is that all of the buds are responding to chilling hour accumulation across all these different varieties in the very same pattern. We see that there's a critical inflection here where about 50% of your deacclimation potential occurs by around 915 chilling hours. And in the Northeast, that happens around January 1st, just as a rough benchmark. And what this means is that heat before January 1st is less of a concern than heat after January 1st, because the buds after January 1st are much more responsive to that heat. All right, we're almost done here. Just to synthesize what we covered, if we want to talk about cold hardiness, we see that there is differences between gene pools and their response, and those differences, the magnitude of those differences change based on the severity of the winter. We also note that between these gene pools that we have differences in this bud break phenology, where some varieties are considered lower chill or some varieties are considered higher chill. But if we integrate the deacclimation data that I just showed you, how they respond to warming temperatures during winter, we come up with a simplistic idea that if you take the cold hardiness and you divide it by the deacclimation rate, can you predict the days to bud break? And when we do this with our data, we see that there's a very nice correlation between these factors with the ability to predict bud break. And so here you see the prediction for days to bud break on the Y and actual days to bud break on the X. And while we want this to be 1-to-1, and it's not quite 1-to-1, this is a really nice correlation between these factors. And this has led us to believe that we can not only predict cold hardiness, but also predict bud break, which is the foreshadowing for Al's talk. So just to conclude, cold hardiness is a dynamic trait. The maximum hardiness of the grapevine is achieved both to genetics and the environment. In some cases, environment is more important than genetics. Acclimation appears to be driven by genetic differences, specifically as they interact with cycling temperatures. So vinifera cultivars respond less than hybrid cultivars to the same temperature cues. And we don't know what the critical exposure is or exactly how to fine tune it is, but it seems that the larger and colder that the temperature cycles are, the stronger the response. Again, as winter's warm, we don't know what this is going to mean, but if you take it as the most simplistic, as things get warmer, it may interfere with proper acclimation. Deacclimation is also driven by genetic differences as they interact with chilling accumulation. I know this is a lot to read, but I'm sure we'll send out the slides or whatever if you want to read it to yourself. As I mentioned very briefly, hybrid cultivars that are derived from Vitis riparian and Vitis labrusca appear to respond to heat and deacclimate rapidly, certainly more rapid than vinifera cultivars, but that there are also hybrids that have been produced through crossbreeding and integration with Estivalis cinerea and rupestris, which seem to be much slower in this response. So this suggests that the deacclimation response is an adaptive trait and perhaps a concern as climate warms. As our winters get warmer, we need to be a little more, we may be a little bit more nervous about some of our cold hardy hybrids in the way that they respond. And then finally, these results have given us the opportunity to build different models which incorporate the different acclimation and deacclimation aspects of grapevine physiology. And we've produced some cold hardiness models that are part of this Cold Snap project, but also lead to potentially bud break models. And so that's what I'm going to give you for the summary today with the handoff to Al, and just do a real quick thank you slide. Al did most of this work when he was here working as a student, and then Dr. Hongwei Wang has picked it up and continued working on it. And Hannah Martins is my lab manager and basically does all of the hard work. And so with that, just acknowledge the grant, other forms of funding, and a whole list of vineyards and collaborators at Cornell that make this happen. And with that, I'm going to stop the share because I think we're doing questions at the end.
Great. Thank you, Jason. That was a really nice intro to cold hardiness physiology. And so now we're going to let Al start his part of the webinar. Just to remind the audience, I've posted on the chat the link to where the recording of this webinar is going to be if you want to go back and read all that information that Jason had, and just to review with more time the information that has been shared.
All right, so I'll go ahead and start now. So as Jason was talking about here, and something that may be intuitive is that cold hardiness is being lost as the plants are breaking bud, as the grapes are breaking bud in the field. And the way that this can be intuitive is you can think of the temperatures that you're experiencing now during the winter. If they happen as the plants are starting to grow, we often see frost damage, and that's easy to spot, is very visible. A lot of the things that we were talking about is damage that is enclosed within the buds, and so it doesn't, because there's no visual markers for it from the outside, it might not be perceived very clearly. And so in here, what this figure is showing in the y-axis, this is just a measurement of cold hardiness, just in an abstract way, the amount of cold hardiness. And this is time under forcing. Jason explained what forcing is. It's when we put these cuttings into warm temperatures and long days in a controlled environment, and we see them losing cold hardiness at a certain rate. And when they finish losing their cold hardiness, we start seeing bud break happen. During the time when they're losing cold hardiness, we actually don't see changes in what the bud looks like from the outside. And then after, we actually start seeing the buds breaking, and then we see leaves coming out. And so this is something we've seen over and over with grapevines. Every single winter, we're trying to do these measurements, and we actually see, if there's people here working on or that have different crops, we actually do see this in many other species as well. And so our question was then, can we use our cold hardiness model or models to predict bud break? Bud break is something that is interesting from a management standpoint. It's something that we can think about damage happening at that time as well. And so knowing when bud break is happening is a good piece of information. And so since we know that we can predict cold hardiness with some degree of accuracy, what does that mean for bud break? And so the first step for that is actually calculating what the cold hardiness is. And what I'm going to show you here are things that we've done with a model that we now call NYUS.1. And that model was created based on some of the studies that I conducted while working with Jason there in his lab. And based on all of those responses, we came up with a set of equations that I won't get into. And this is just a visual representation of those equations. This is what the model looks like from the calculation standpoint. And as he mentioned, there's acclimation and there's deacclimation that are built into that model. And so cold hardiness at any time is a response to temperature. And how cold hardiness is achieved is the result of this phased integration of acclimation and deacclimation. Its acclimation is a little bit stronger in the beginning of winter. Deacclimation starts becoming stronger and stronger, which is something that Jason presented, something that we know is a response to chilling accumulation. And it's this chilling accumulation that does the handoff between acclimation and deacclimation. So the same temperature during the fall can during the fall promote acclimation, but during the spring it'll actually promote deacclimation. So there's this switch that ends up happening over time. And so what this model looks like when we're looking at temperatures over the course of a winter, and soon it'll make sense why this is showing the winter of 1983 to 1984. We see that based on air temperatures, we can predict what cold hardiness looks like. Cold hardiness being here the blue line, the gray part is the temperature up here, the air temperature. And we see that acclimation period over here. We see sort of a maintenance period in the middle of the winter and the deacclimation over here. Something else that happens sometimes is reacclimation. We can have instances of reacclimation, which is an important aspect in how we end up predicting bud break. That maybe some of you have heard of what growing degree days are, and growing degree days sometimes are used to predict bud break or to think about when bud break is going to happen. But something that is interesting of thinking of cold hardiness is that reacclimation is something that we know exists in the buds, and we can calculate that. But growing degree days only move forward. You don't have negative growing degree days. And so this might be where using cold hardiness can be a better way of thinking of when bud break is happening. So we've calculated our cold hardiness, and then we start relating cold hardiness to bud break. And I'll explain why there's these two ends to the curve, and hopefully it'll make sense to you. So in here, the curve actually has a 10th, 50th, and 90th percentile. So we're trying to think about the population of buds that are in the field. And something that is happening here is that we're letting the model run into positive values of temperature, positive in the degrees Celsius scale. So this would be above 32 Fahrenheit, which doesn't make sense for if you're thinking of cold hardiness. Cold hardiness is always in that in that sub-freezing range, right? And so we let this model continue running up, and it continues accruing these units that are in degrees Celsius. When in reality, we do apply this threshold for the cold hardiness that is based on some work, some observations that we've actually realized as we were doing this work that are quite old, actually. And perhaps need to be reviewed, but they're the reference that we have for what are the temperatures after all cold hardiness was lost of those tissues that are beginning to grow. Okay, so this is the separation. But then we have this threshold that is applied up here that is the cold hardiness at bud break. And it's a cold hardiness that doesn't actually make sense from a value standpoint. Here we're looking at 5 degrees Celsius. But it makes sense for our modeling purposes. And when our line crosses that, we actually predict bud break to happen, okay? And so this is what our overview of the project is in a larger context. For simplicity, and I'll try to actually cover everything. I've actually removed some parts of this that maybe would not be of interest for those that are not as inclined to like math like I am. So this is the part that we'll actually see in here. So the first question that we have is, can we predict bud break? And so what we did was we gathered this phenology data set, which means that there's a lot of people that collect data of what was the date when plants started breaking bud, or when 50% of the vines were breaking bud. And we got these data from 3 places in North America, 2 in the US, Portland, New York, Lewisburg, Pennsylvania, and Niagara-on-the-Lake in Ontario. And then we have 4 locations in France and 1 location in Germany, where they've actually been really good. It's over 50 years of data in many of these locations, which are very helpful for us. And we have 3 cultivars that we're looking at: Concord, Riesling, and Cabernet Sauvignon. So the first question is, can we predict bud break? And actually that ended up being somewhat of a simple question. Here is a graph showing the observed day of the year when bud break happened, and on the Y is predicted day of the year. So if we had a very precise prediction, we would have this line be a perfect diagonal over here for our data. All our data points would fall under that line. And it turns out that our data actually, it looks good. Our predictions are quite good in the metrics of how we evaluate these things. One of the ways that we evaluate this is using the mean square error, root mean square error. And this being around 7 means that we're predicting usually with 7 days of error is what we're doing. Sometimes we're really good at it. Sometimes we're doing a poorer job. Like, for example, these points that are quite out from where they should be. But a lot of them are falling within a narrow interval of when bud break actually happened. So the first answer to that is yes, we can predict bud break using these cold hardiness models, okay? But there's a lot of interesting questions that we can now start asking since that model seems to work and work across these very different environments where we're doing this. So first is an exploration of uncertainty, of like the error that we find in our predictions. Why are we sometimes really off? And one of them is maybe because of freeze damage. And so let's take a look into freeze damage here. So these are our predictions when we're looking at all of the predictions combined. So our 329 time points, and they're centered at 0. And a lot of our predictions are falling into this narrow range here. You can see that between This is the error. So how many days away from the date when bud break was observed are we looking at? And so a lot of our predictions are very good and falling within 10 days of when bud break actually happens in the field. Something that we observe is that if we separate the years when we're predicting damage to occur to the buds, we actually are predicting early. You can see that if the points fall in the blue side here, we have an early prediction. And if they're in the orange side over here, we have a late prediction. And so when the buds are damaged or we predict some damage to occur, we're actually predicting bud break to happen early. Okay, so we want to look a little bit into that. To look into that, we have to think about safety margins, and I'll explain what safety margins are. The safety margins are the distance between the cold hardiness and the minimum temperatures that is observed. That cold hardiness shifts throughout the winter. Sometimes it's really tiny, sometimes it's larger, and it tends to become really small as we approach bud break, okay? And so if we see here actually in this year, this is 1945 to 1946 winter, we can see that the low temperature actually crossed our cold hardiness. And so these are instances where we actually identified damage to occur to the buds, okay? And so this is something that will be important in how we are trying to fix our predictions based on cold hardiness. To validate whether damage had occurred or not in our predictions, for Portland, New York, we went back into the Erie Daily Times, which is a local newspaper, and looked for instances where they were mentioning that grapes were damaged. And then starting in 2007, we actually consulted Cornell's extension records, and we could see where damage was reported or not to the buds. And it turns out that we actually predict very well when damage happens. And this is an example here. So in 1945, 1946, we had that spring where plants were damaged quite a bit. Okay, we have a number of instances of damage appearing. When we look at 1946 to 1947, there was, note that it crossed the threshold of the 10% line, but it didn't cross the 50% line, okay? So we predict a little bit of damage in that year, but not a lot of damage occurring. And when we look at a newspaper article from 1947, it actually says that there was no danger of frost, and that if it was development was delayed in grapes and other fruit crops because of the consistent cold weather. And there was no damage compared to the huge loss incurred in 1946 by the area farmers. So you can see here that our model is actually predicting something very, very good compared to what was observed in the field back in those years. And so predictions of damage actually reflect historical reports in Portland, New York. So now, what do we do with this? If we are seeing that some of these buds are being killed, we can actually shift our prediction of when bud break happens. So this is the original prediction of when bud break happened in 1945, 1946. And then because of that damage, we can actually shift it into the 50% of the buds breaking from those that were not damaged. And what this means is that bud break happened 9 days later than what we originally had predicted if we don't acknowledge that damage happened to the buds. It turns out that when we acknowledge these things, our predictions improve for the damage. Here in gray are the corrected predictions. And you can see that there's a shift. The RMSC, like how wrong we were before for the ones that are damaged, was 7.4 days, and it drops down to 7.1 days, which seems like a very tiny change, but it's an important change in there. Okay. And then when we look at the entire group combined, our predictions become better. So we know that if we acknowledge whenever damage happens to the buds, we actually get good predictions of bud break. So it is something that we continue working on with incorporating these freeze damage events. So let's think of some additional implications, particularly as we think of safety margins and the risk of frost to the buds, not just as they're breaking bud, but actually during the entire season. So here in this graph, I'm showing you the probability of damage through the cold season, and this is just for Portland, New York. And in red are instances where we're predicting that safety margin to be negative. So actually like some damage to have occurred. And in yellow is when that safety margin is small. It's close to 2 degrees Celsius. So how often does that happen? This here is data for Cabernet Sauvignon. These are the dates during the season, during the cold season, and they're centered based on 0 being the date when bud break happens for each one of the cultivars that we'll see. So for Cab Sauv, the mean bud break date for there is May 20th, okay? So something that we see is that there's a decent chance of cold damage happening always in the middle of winter for Cab Sauv, okay? But then when we're getting close to bud break, it actually doesn't have that much chance of damage occurring. Here, now we're looking at the 2 other cultivars, Riesling and Concord, where Cab Sauv and Riesling are vinifera, and Concord is one of the hybrids that Jason was mentioning, which is a faster to deacclimate. It breaks bud earlier as well. You can see here that the average date of bud break for Concord is May 7th. For Riesling is May 13th. So Riesling is sort of an intermediate between Concord and Cab Sauv. Now let's look more at the damage part. Cab Sauv has a high risk of winter damage, but less risk of spring damage because of the late bud break. It loses cold hardiness slowly and breaks bud later, and so it doesn't have a lot of risk during the spring. Riesling is somewhat of an intermediate. It doesn't have a lot of risk in the middle of the winter, and it gets close in the spring, but not a lot of damage actually occurring over that time. And Concord has very little, it has a lot of cold hardiness, a very low risk of winter damage of middle of the winter. But when we look at spring, the possibility of spring damage, it actually can be hurt quite a bit by late spring frosts because of that, particularly because of that early bud break, faster loss of cold hardiness. So this is through the season we can try to evaluate that, okay? Now, this is thinking of probability of damage depending on the seasonal temperature. If we look at the average temperature across the entire season, here we're thinking of the dormant season temperature, how often do we see damage happening? And here we're looking at days with freeze damage risk. How many days during a winter do we calculate that safety margin will become negative, that we predict damage to occur? Here again is Cab Sauv first. In the x-axis, we have the mean dormant season temperature. So this is just averaging out the temperature from November until April for multiple locations, and we'll see them soon. And this is the day, the number of days with freeze damage risk. So we see here that Cab Sauv has higher risk when the temperatures are low or places that have a mean dormant season temperature that is low. And then it drops as temperatures are higher. there's this little bump before it goes down to 0, where in places particularly where frosts do not occur, okay? Here are the other two cultivars. Where it's very interesting to see is Concord here, that even when temperatures are very low, in places where temperatures are very low, the average probability of damage is actually quite low. And it increases as temperatures increase because it responds so much to warm temperatures that it breaks bud too fast, and it's very prone to being damaged by spring frosts or late frosts, okay? Riesling, again, an intermediate, it suffers some damage at those low temperatures. It also has that bump, but not as high as Concord, okay? This is now if we look at all the climates together that we were evaluating, and Portland, New York is around this range, okay? So I'm gonna draw it down here. And we can see that if we were to average out this curve, it would sort of be a flat line because it's decreasing in the lower range, and then it's increasing over here. It seems to have the grapevines seem to have this sweet spot where the winters are cold enough that the plants remain dormant for a very long time, and they break bud late enough that they won't suffer from late frosts. Here, the plants are gathering damage from late frosts. Here, the plants are taking damage because of the low temperatures of the winter. Okay, so depending on what climate the plants are in, this will vary. So I'll just go fast through this. Bud break sensitivity. This means what day of the year buds are breaking. If temperatures are warmer, you're breaking bud earlier in the season, but that rate sort of changes depending on where we're at. And one thing that comes across as very important for us as we're doing this, and this is a very technical slide, just to say that chilling accumulation is a big question for us. Jason mentioned that there's those hours that are counted in chilling accumulation. And we don't know if the models that are predicting that we know that they're not very good, and we're trying to figure out what to do with them. We've had work on that where we're trying to improve these models, and we have yet to incorporate these new models into our predictions of bud break and in our predictions of cold hardiness. Okay, but we know that chilling accumulation is a problem. So the conclusions for this part is that we can account for bud damage from cold, and that improves our predictions of bud break. There is a cultivar-dependent pattern of damage risk to the buds and to growing tissues. And the locations differ in the patterns of freeze risk. Some places might be seeing, with climate change, they might be seeing more instances of damage. Some of them might be seeing less instances of damage. And that all depends on where you are in that scale of mean dormant season temperatures. So it's a very complex problem, and different locations will observe different effects as temperatures change. Phenology advances, it happens earlier, so bud break happens earlier with warmer mean temperatures. But there's a point where this changes, and I don't have time to go into that, but if there's people from warmer climates don't know that lack of chilling is a problem. And now I'll introduce what cold snap is, but we have this project going on where we want to understand chilling better to improve all of these models, okay? But in general, using cold hardiness to predict bud break actually gives us some very accurate, some accurate and precise predictions of when bud break is happening, but it also allows us to do so much more related to the cold hardiness itself and the possibility of cold damage. So now introducing ColdSnap very quickly is a project that we have going on where we want to validate and expand these cold hardiness models. And the other part is that we're creating an online tool for cold hardiness monitoring for stakeholders to access very easily this information, depending on where they're located. This is the part that I've presented a little bit on, and this is what I'll talk a little more in a couple slides. But this project involves a lot of people, and I want to take some time to just thank the people that are sending us material, grapevine material, so that we can measure cold hardiness. We can study chilling a little bit more in depth. And this is happening across a large gradient of climates across the U.S. This is overlaid on the USDA hardiness zones map, so that you can have an idea. So thanks everyone who have contributed either data or they're actually sending us material. Okay. As for the tool, it's not out yet, but this is what the website is going to look like. You're going to be able to select your weather station and what cultivar you want to get your predictions for. It'll show you visual results, and then it also shows a forecast of whether there is risk or not in the near future based on weather forecasts. If you're interested in learning when this tool comes out, it should be within the next couple of days. You can use this QR code. You can it'll ask for your email, so we can send you an email when the tool is published. Also, something that might be important, the tool is formatted in a way that you can use your phone. It'll be easily accessible on your phone to get this information. Okay. Something that I'll mention as well is that Hongray, who works with Jason, also has another website where working with which houses many of these models as well, and where you can obtain some cold hardiness data too. And if you look up VineCold Cornell, you'll actually get to that website. And maybe Jason, you could post a link to the chat as well. Okay. And with that, I just want to thank a lot of the people that are involved in this work. Erica and Francisco are the students that actually took the modeling really forward to the point where we're predicting these bud break data. Mike North is the person that has been managing all the ColdSnap data collection. Jason, Hannah, and Hongray, Dan Olmstead working with getting the website in place, and other people that have contributed bud break data into these predictions, and some people that have reviewed our work. And thank you for your attention and questions.
Thank you, Al. Could you go back to the slide where you have the QR code, so people that are interested on getting some information about when the tool will be available can just fill up that Google form, and we'll let you know when the tool is live. This grape cold hardiness risk assessment tool will be available through NEWA. So those of you that are familiar with NEWA and some of the other models that they have available, you can it's going to be sort of very similar to the other models that they have there, which you just go to the website, select your preferred weather stations, and you are able to run it. And Jason provided a link there in the chat to the other tool that is already available through his lab. So there are a couple of questions on the chat that Jason was able to answer, but there's here a comment about being able to predict bud break consistently just using growing degree days. So I just wonder if Al, this is something that you want to address.
Sure. Yeah. So growing degree days are a really good tool to use, and we see them as perhaps like a simpler way of assessing when things might start growing. They have problems with them, where this might be very technical, but there's a linear response in growing degree days that we can't actually detach ourselves from them in how we calculate these growing degree days. It's also the fact that we can't go backwards in them. As I mentioned with reacclimation, it is a problem. So it's actually a good something that can be mentioned is that when we're using growing degree days, that they can actually be pretty good if you're just looking at a single location. So for your location, you might observe bud break within a certain range of growing degree days every time. But if you're trying to see this across multiple locations, we start losing this relationship, and that's a part of the problem. Is that as climates shift, you might actually have to shift the number of growing degree days that you have, and we're trying to find a physiological explanation for this, and have a model that is independent of what the climate is. We can always predict bud break.
I'm gonna leave a couple of minutes. If anybody else has some questions, you can just go ahead and post it in the chat. In the meantime, I'll just post here again the link of where we're going to have the recording of this webinar. You want to go back to watch this webinar? I know it was a lot of information, and just also tell everybody that we will probably be having another webinar in the future, in which we're going to introduce the grape cold hardiness risk assessment model, and just go through how it works. So there's a question from Beth. Yeah.
Oh, sorry. I was going to say, I have a question that was sent directly to me that I can go over after.
Okay, so you can go over it now, or
I'll just do it quickly now. So the question is that we've been seeing a lot of unseasonal warm temperatures happening, or like false springs that happen in the middle of the winter and late winter or earlier springs, and what is happening with the rates of deacclimation. This is actually something we've looked at, and what happens is Jason was mentioning that if these warm spells happen early in the winter, plants don't have a lot of issues with that. They won't lose in the same way. It's when they start happening later and later that these become problematic, because the amount of deacclimation that will happen will be quite a bit during that warm spell, because the deacclimation rates are higher. And reacclimation late in the winter and in spring are actually quite slow. They're very complicated to happen. We don't know why, but that's what we observe. And so it can be very problematic in that way. But the models do acknowledge that as happening. Yeah, sorry, Amaya.
No, that's great. Thank you. So there's a question here from Beth, and I assume it's related to the previous answer for the growing degree days question. It says, do you think this is a feature of reacclimation, or speculate other things too, and more of a problem applying it in warmer climates?
Yeah, so this is thinking of the reacclimation. Yeah, so the negative growing degree days, that would be an instance of reacclimation. Yeah, and this might be a part of it is a conceptual issue of what growing means in terms of plants. Growing is an irreversible process. So trying to think of negative growing degree days is not something that you want to do in particular. But also, if we can predict both things at the same time, maybe that's better. So I that. And is it more of a problem of applying in warmer climates? Warmer climates actually have it is because This is a climatology question where in very cold climates, once it starts warming up, that warming usually is very consistent. Um, in the season, day length is very long. You get a lot of sunlight, so it's the warming happens in a faster way and very consistently. In warmer climates, the change of season actually happens in a much slower way, and you have a lot of warm temperatures in the middle of the winter. So selecting from when you're counting your growing degree days can actually be a very non-trivial question. And so that's where, like, sure, if you're looking at small areas or a single location, growing degree days could actually work very well. But if you're trying to do this for a larger region, then it starts becoming very problematic. I don't know, Jason, do you want to add anything?
No, I'm good. I'm also trying to answer some of the ones that are popping up in real time.
All right. So there's a couple of other questions here. Kevin asked, does the date of when injury occurs affect bud break date? Midwinter versus late winter versus close to bud break?
Oh, that's a great question. It and at some point we had a figure that was showing this. It's when the damage happens in the middle of the winter, there's very little change in when the bud break is actually going to
Mhm.
happen. When the damage happens close to bud break, um, with a certain distance, it delays bud break. If it happens right before bud break is set to occur, there's very little change actually, and it seems like uh, some people might have experienced this where the buds do get damaged, but they sort of like push out of that envelope. They continue and they just like put out this growth that is dead, or the buds look dead. And that's what we tend to observe in terms of the data in how we're predicting things.
Well, that was actually one of the things. I think the perception of the bud break delay is also in part because the buds that are breaking is a distribution of break time. And what happens is
Mhm.
when freeze occurs close to spring, it kills the early half of your distribution. So then your visible bud break distribution shifts to what would have been the lazy buds or the old, like the buds that were a little bit further behind. And so you get dis, this dissonance between when bud break would have been predicted and when we saw it, mostly because you cut half the distribution. Or I don't want to say mostly because again, I'm not. My understanding of it is that you break that distribution enough that you shift the population mean, and so then if we want to, when we're doing these models, being able to predict when the model fails because damage occurred is actually validation that we think we know what we're talking about. If that makes sense, because we modeled the ones that were killed and removed from the population, so the mean shifts. A little bit complicated, but it's not that the freeze damage causes partial damage to the bud, right? It's most like when you go past the LTE, it's pretty much an all or nothing.
Yeah.
I have another question from Hunger saying, how did you determine the threshold cold hardiness at bud break, the 5 degrees Celsius in the bud break model? Does this value differ between different cultivars?
Yeah, so that was determined based on experimental data that we had from growth chambers. I actually I had that slide in the presentation and then I ended up removing it. But it's at the same time as we were measuring cold hardiness loss in these forcing assays, we were looking at bud development in another subset of buds. And then we could just correlate the cold hardiness where the cold hardiness curve was in those lines that are linear with when we observed the EL stage 3, which is what we were using. That's the phenological scale for grapevines. It's a bud break measurement. Yeah. And they do differ between the cultivars, and it's something that's based on data. It seems like a lot of the hybrids they break bud in a slightly different way than vinifera does. Vinifera has some more room inside of the bud, so the tissues have to grow a little bit more before bud break is observed. Whereas the hybrids, they have buds that are very tightly packed, and so any movement, they already start showing bud break.
Okay, so we have another question here from Greg. It says, please re-explain grapevine bud break from dormancy with reference to selecting hardwood cuttings relative to endodormancy versus ecodormancy. Sounds like it is best to collect dormant cuttings in late winter rather than at the beginning of the winter. And I would assume that based on this question, these are cuttings for propagation. Maybe Greg can try to explain why his questions about why he's interested in collecting cuttings.
I sent him a response back. So in case he doesn't say it, I think I asked just basically if he wanted to know when you would get like the most push, like the most efficient sort of collection to push. And then I stated ecodormancy, late winter is the better collection time. If you collect endodormant wood, you could certainly propagate it, but because it would have to satisfy sort of the chilling, you basically, it depends on your propagation conditions, but if you're bearing sticks basically in moist soil from early winter versus late winter, the rot versus propagating balance might it would be in your favor if you did late wood, right? The longer it sits in the trying to chill in wet, moist conditions, you're also encouraging fungal growth and all kinds of stuff. And so I would say efficiency probably in the ecodormic phase.
Good. Then the last question we have is how to measure, I assume it's growing degree days.
There are a number of calculators out there. It's basically a summation of the number of days or hours above a certain temperature threshold. It's actually a tool I think that we need on NUA, and maybe there is one on
it is
NUA.
available.
Yeah, it is
It is available on NUA.
So if you have a NUA site, you could certainly go use that tool. Or if you have a NUA station and you're on the NUA site, you can go use that tool. I don't know if there's online calculators that will do it just based on a map pin, but typically that's how you do it. Just use an online calculator.
Yeah, I think the easiest way would be to try to use NUA and find, if you have a weather station that is connected to NUA, you will be able to find in their menu there how to calculate growing degree days. And I think that you can adjust the base temperature for that calculation as well. And you can use, if not, you can use a weather station that is close by. You can also just Google a calculation of growing degree days. I mean, it's a simple formula you could actually import if you have weather data from your site. You can just import it into an Excel file and just put the formula there and just calculate it. But I think at this time, going through NUA would probably be the easiest one to do. Okay, a couple more questions. I think this is going to be the last two questions because we are almost 15 minutes past the hour. Thank you for all the questions. Knowing bud position on cane can impact bud break as well as observed bud hardiness survival. Does training system have an impact on prediction as does pruning strategy to delay bud break? So bud position in the cane and then also pruning to delay bud break.
Yeah.
Go ahead. You can go.
Okay, I can talk a little bit. So in particular, in the middle of the winter, we've done some measurements, and it seems like at that time, the position of the buds do not matter. We've done this was while I was at Cornell still. Very, very long canes of Concord with hundreds of buds, and we didn't see differences across that range, which was quite interesting. it does seem like in the spring, this is something that happens depending on your training system. If you're in a location with low chilling accumulation, also, the buds at the tip of canes will start breaking earlier. And it is something that, like for pruning, actually, Amaya does that in the vineyards here, where you prune long, and then after the tips start growing, you go back and prune again, so that you delay bud break for the lower buds, which is an alternative. The difference in cold hardiness between those is very slight, but it is helpful because that time when buds start growing, when you have the first few leaves out, is the moment where the plants are the most cold sensitive of the entire season. Even after, once they start growing a little bit more, once the leaves are more developed, uh, they're less sensitive to cold damage than they were at that very moment when the buds start breaking and leaves developing. So yes, it's a minor difference. We don't acknowledge that in the model very well because it's so hard to do.
And then
The
the
only
last
thing,
question.
well, the only thing I wanted to add onto that is that within vineyard microclimates and within region microclimates can have a really big effect, even more, I would say, than a pruning system difference. And so when we are doing cold hardness modeling, I think our comfort is like within 2 degrees. I think 2 Celsius, either direction, is sort of like where we've centralized. I have studies looking at microclimate effects on a couple different cultivars, and we can get up to 5 degrees variation between the least cold hardy and the most cold hardy vineyard of the same cultivar, same rootstock, same clone. None of those factors matter as much as microclimate can sometimes matter. And so, yeah, just wanted to add that in there.
Right, that's a substantial difference, 5 degrees. All right, so the last question that we have is about hydric stress effect on when ecodormancy starts.
I would say that we've never done experiments with hydric stress as it relates here, but hydric stress is used in some systems to sort of trigger a chilling hour effect on the dormancy transition. I think Al can speak this better in the tropics, but there's lots of different stresses that basically anything that creates sort of like a reactive oxygen burst within buds can help transition them from endodormancy to ecodormancy, and drought can play a role in that way. I can't say that it's as effective one way or the other, but it can help fulfill some of that.
Yeah, we worked with some data from was it off of California from Krista Shelley, where they applied stress during the growing season, and it affected dormancy. It seemed like it made plants go to a deeper dormancy later in the year and actually come out of dormancy earlier, so a shorter period of dormancy. So there's an effect, and where Jason mentioned that, like I could talk about in the tropics, is because I'm originally from Brazil. There's an area in the northeastern part of Brazil where they grow grapevines, quite a bit of grapevine, and they do some of the dormancy there is through because the temperatures never drop below, I think it's like 60 Fahrenheit. And so the dormancy is through water stress. So they withhold irrigation. It's a very dry region, and then when they start irrigating again is when plants resume growth. Irrigation and application of hydrogen cyanamide as an alternative. So yeah. Water stress has an effect, also not like should in some way be incorporated into the chilling models, but we're still a little bit limited in there.
All right. Well, I think we answered all the questions, and so just thank you to Al and Jason for a very, very, very interesting and informative webinar. stay tuned. We will probably have, as I said, another webinar on the grape cold hardiness risk assessment tool that is going to be available in NUA shortly. And thank you everybody for attending the webinar today.